we're going to be talking about macromolecules, carbohydrates, and lipids. I'm going to break it into two videos. So first we'll focus on 2.1, which is about macromolecules in general. And then the second video will be 2.3, which will be about carbohydrates and lipids. So if you look at this meal here, you can see lots of different types of foods. And it is packed with macromolecules. So we're going to get into that more specifically in this lecture. So you could pause the video for a second and try to think to yourself, what do we use carbohydrates, lipids, and proteins for in our bodies? So hopefully by the end of this lecture, you'll be able to answer that. So the first understanding is molecular biology explains living processes in terms of the chemical substances involved. So when we look at this whole unit, we're going to be talking about parts and then the outcome you get from those parts. So they say that biologists study things from a reductionist approach, which is basically reducing the outcome down to its pieces that make it happen. So we look at all the different parts to try to understand why this occurs, and in the sense of why we get this emergent property from, for example, maybe we could say with DNA, from different nitrogen bases and sugars and phosphates. Why do we get this genetic storage device? Some scientists think that the reductionist approach alone ultimately is going to limit us because some molecules have dual roles. And so even though we're looking at one outcome that this molecular structure does, it might do another one. And so we need to look at it from that pathway as well. So it can be a little bit limiting when we're trying to break down one outcome into its parts. So understanding 2 is about carbon. Carbon atoms can form 4 covalent bonds, allowing a diversity of stable compounds to exist. Carbon is the most important element when it comes to living things. And even though it's the 15th most abundant, so it's not the most, but it's 15th most abundant on our planet, it forms the backbone of every single organic molecule. And the reason is because of this. If you look in this picture here, you're going to see the outer valence only has 4 electrons in it. So that means it can bond with, each of those 4 can bond with another element that needs a partner to create covalent bonds. So that allows it to have 4 bonds that'll make it be a very stable 8-valenced outer shell, because all molecules want to have 8 electrons in their outer valence, and that'll make it more stable. So carbon has the potential to make infinitely a number of different possible molecules that are going to be very stable, hence why it's the main structure of living things. I want to go back to this. So if you look here, this is methane, and you can see that the orange dots are just carbon's electrons. But then if you combine it with hydrogen, which only has 1 in there out on electron, it's dying to bond with and share that space with carbon because it'll be more stable. It'll have 2 in its one ring, because there's usually 2 electrons in the innermost ring and then 8 for any rings after that. So this makes hydrogen happy, and then it makes carbon happy, because now it feels like it has 8 and it's stable, and hydrogen has 2 because it only has 1 valence ring. Okay, so organic compounds contain carbon and are found in living things. So here you're going to see different types of sugars, and you can see that the ring is all made of carbon. So pay attention to where there's a C in all of these different structures. So here are sugars. You can see tons of carbon. Amino acids, the main structure is carbon. This is a lipid here with a glycerol head and a fatty acid tail. You can see the tail's center backbone is made of carbon, and then so is the glycerol head. So usually the carbon's bonding with hydrogen or maybe another carbon is what we're going to see in these molecules. Notice the word organic compounds is highlighted. Anytime you see that word organic compounds, it's talking about a molecule that has carbon. And all living things are made of organic compounds. It's usually involving a part of a living thing, or it came from a living thing. Functional groups. Functional groups are important in chemistry because they are the portion of a molecule that is capable of characteristic reactions. So we're going to be talking about metabolism in a bit, and how these macromolecules form because of metabolism. But when you look at the molecules, you're going to see these parts here are called functional groups. Okay, a hydroxyl, a carbonyl, carboxyl, amino, and phosphate. And they are the group in each of these macromolecules that will react with another carbohydrate or another lipid or another protein. So when proteins bond with each other, it's the carboxyl group that's going to be bonding with each other. When lipids bond with each other, it's the carbonyl group. So if you can memorize these, you'll know what you're looking at as soon as you see a structure. If you see a negative OH coming off of a structure, you'll know that's where of a carbohydrate, which you'd recognize by a 6-carbon ring. You'll know that that's going to be where the bonding takes place. So if you memorize these, you'll know exactly where you would need to put a bond if you were asked to do a reaction of a structure. Just so you can get used to some of the symbols you're seeing, when you see one line like this, that's considered a single covalent bond. So not an ionic bond, remember, a covalent bond where they share electrons. And when you see 2 lines like this, that means it's a double covalent bond. So think of a double bond as being a stronger covalent bond, and then a single bond's a little weaker, but still covalent bonds are the strongest. So just so you recognize the symbols. So understanding 3, life is based on carbon compounds, including carbohydrates, lipids, proteins, and nucleic acids. So we're going to be talking about these 4. These are the macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And that's what all life is based on. So let's start with carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen, so CH and O. Remember, CHON, so it doesn't have nitrogen of those 4 main elements. Organic compounds consisting of 1 or more simple sugars are considered a carbohydrate. Monomers follow the general basic formula of CH2OX. Okay, so this is, if you look here, it's part of the glucose structure, CH2O, and then X is meaning there can be as many as possible there. It's like a squaring something. Monomers are commonly ring-shaped molecules. So you will always see with carbohydrates this carbon ring right here with carbons on each of these points. These are some examples of different polymers of carbohydrates. So glucose is the monomer, okay? But starch would be one that often would be, it comes from plants. And notice that when it's all bound together, so it's a very long chain, it's a polymer, it's moderately branched, so it's a little bit less tightly branched. Glycogen, which is our main energy storage in humans, is highly branched. Oh, and starch is also a big energy storage source for us. And then there's cellulose, which is unbranched. Cellulose is actually one of the carbohydrates humans cannot digest, so it goes right through us, but it's in plants. And because it's unbranched like this is part of the reason why we can't digest it very well. But these would be 3 different polymers that are carbohydrates. So the next one is lipids. So this is a picture of a monomer of a lipid. So you can see there's the glycerol head and then 3 fatty acid tails. Notice that there's carbon throughout the tails and the glycerol head. So this would be called a triglyceride. Lipids are a group of organic molecules that are insoluble in water, so they don't dissolve, but soluble in other non-polar organic solvents, like ethanol, for example, could dissolve a fat. Okay? Common lipids include triglycerides, which can be fats and oils. So fats you would normally notice are solid at room temperature, and oils are usually liquid at room temperature, but they're both a type of triglyceride. Also phospholipids, which are part of the cell membrane of the cell, as well as organelles membranes, membrane-bound organelles, and then steroids are also a type of lipid. Proteins are extremely important in the human body. They contain carbon, hydrogen, oxygen, and nitrogen. Additionally, sometimes sulfur is common, but it's not always present in the proteins. Proteins are large organic compounds made of amino acids, and they're arranged in one or more linear chains. So this is a type of amino acid, if you remember from 10th grade. Leucine is just one that you would find on a codon chart. There's 20 different amino acids that can be put into amino acid chains based off of what the DNA codes for. And these amino acids then form a chain, which then folds into a special shape based on their attraction to each other. And that shape then determines that protein's function. So different types of proteins are hormones, enzymes, immunoglobulin, which is a type of antibody in your immune system, as well as gas transport. Hemoglobin is in your red blood cells, and it's what carries the oxygen. So very important proteins. So the monomer is amino acid, and then the protein would be the polymer. So life is based on, the last one would be of a carbon compound, would be nucleic acids. So nucleic acids would be like DNA and RNA. They contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. The chain subunits are called nucleotides. So if you remember from 10th grade, a nucleotide is a nitrogen base, a sugar, and a phosphate. And grouped together, they are the pieces or the monomer that make up nucleic acids. In a nucleic acid, if the sugar is ribose in the nucleotide, we know we're looking at a ribonucleic acid. And if the sugar is deoxyribose in the nucleotide, we know we're looking at deoxyribose nucleic acid. So here's an image of one nucleotide here with a phosphate, a sugar, and a nitrogen base. So if we're going to talk about macromolecules, we have to talk about how they're formed. So the fourth understanding is about metabolism. Metabolism is not what you always think of. I think most people, when they think of metabolism, they think of their food being broken down, which is a form of metabolism. Actually, that would be catabolism. But metabolism is like the main category. And there's two, it's basically any enzyme catalyzed reaction in a cell or in an organism. So it could be the building up of something or the breaking down of something. So any chemical reaction occurring in your body, we would call metabolism. And it's super important. All living things have to be able to do this. So there are two pathways that metabolism can happen, which this would be animalism, which would be to build or synthesize complex molecules from simpler molecules. So this would mean taking monomers and bonding them together to make polymers. And the reaction that does this is called condensation, and we'll talk about that more in a bit. Catabolism would be the other pathway, which is the one you most commonly think about when you think of digestion of your food, is the breakdown of complex molecules into simpler molecules. So taking polymers and breaking those bonds and making them back into just monomers. And we call the reaction that does that hydrolysis. So metabolism, remember, is all enzyme catalyzed reactions in a cell or organism. And why I say enzyme catalyzed means that the enzyme is needed to make something either build itself or break it down. And catalyzed means it makes it go faster. So the enzyme will make this happen in a slower amount of time. In a faster amount of time. All right, so there's two different understandings here. One is about animalism, and one is about catabolism. So let's first look at animalism. So anabolic reactions are those which build molecules. So for example, protein synthesis, where we're taking the amino acids and building it into a protein. Okay? Condensation does this, and condensation makes bonds, and you could think of the word condensation as water releasing, and I'll show you what I mean in an animation here in a second. But if you look here at this condensation picture, you're going to see this OH and this H here. Those are going to be removed, and this oxygen is going to be left to create a bond between these two carbohydrates, or glucose is what they are, and it'll create a glycosidic bond here. The other type of reaction is called hydrolysis, which will allow catabolism to occur. So remember, catabolism reactions are those which break down molecules, for example, digesting of your food. So whenever you see that word lys, it means splitting, okay? So hydrolysis, hydro, water, is going to be water splitting. So water is actually going to break the bonds that were in place in the polymer. So in this picture here, you're going to see there's a bond here where there's these O's in the hydrolysis reaction, but if we add an OH and another H, it's going to kind of return back to what it looked like in this end condensation reaction picture. So it's going to, by adding water, you're going to keep those bonds basically satisfied because they're now stable with an OH, a hydroxide, and another hydrogen. And it'll split that bond. And then you'll be back to monomers. So let me show you the animation for this. But just to remember that what's doing this, all of these reactions I'm talking about, animalism and catabolism, or an anabolic reaction and a catabolic reaction, require an enzyme to do this, to remove the water or to add the water. And it's going to make the reaction happen faster. So let's watch this animation.
A condensation reaction joins two molecules together to form one larger molecule. An enzyme removes a hydroxyl group from one molecule and a hydrogen atom from another, then speeds the formation of a bond between the two molecules at their exposed sites. Typically, the discarded atoms join to form a molecule of water. Hydrolysis is a type of cleavage reaction. It's like condensation in reverse. An enzyme splits the molecule into two parts, then attaches a hydroxyl group and a hydrogen group from water to the exposed sites. The result is two smaller molecules. Condensation and hydrolysis are used to put together and break apart many biological molecules.
Okay. So you could see in that animation how anabolism happens and catabolism happens. Okay, so just to review, condensation is along the top, hydrolysis is along the bottom. So condensation, you're going to see monomers, two separate monomers, take water molecules out, and then they're going to bond together, and you're going to get a dimer, or it could be a disaccharide, for example, because there's two of them, so hence the di. Then the dimer is going to then take another water out, and it's going to bond to a third one, and you're going to get a polymer. Right? Anything more than two is going to be a polymer. On the other end of things, if we were going to go backwards, we'd have a polymer, and if we put a water molecule in, you are then going to split them apart, so you're going to be left with a dimer. And then if we were to take water out of this dimer, we're then left with two separate monomers. So there are enzymes like synthase that's doing this, and an enzyme hydrolysase that's doing hydrolysis. So this table just kind of sums up what you're going to get from the different processes. So I'll let you review that, but just so you can see, this is the condensation reaction column, and this is the hydrolysis column. So if you need to pause it to look at this, you can. What I do want to point out to you here is this is the different types of bonds that form. So when it's building proteins, it's a peptide bond. When it's a carbohydrate, we call it a glycosidic bond. And when we're building polymers of lipids, it's an ester bond. There's always water as a waste product from the reaction, since water is an OH and H are removed to make that bond. Okay, so just to review so you can see the chemical reaction. So this would be an example using to make polysaccharides. So we have a monosaccharide, which is a sugar, okay, or are the monomers of a polysaccharide. So usually it's glucose. So a glucose plus a glucose, here you can see the H coming off of this carbon and an OH coming off of this carbon. We're going to remove it. We call that reaction condensation, so an enzyme's removing it, and that's going to create a glycosidic bond, because carbohydrates, we call that bond glycosidic. And then notice at the end, so these two glucoses have bond together, and at the end this is a water as a waste product. So in this case, we had 2 glucoses, and through a condensation reaction, it made maltose, which is an example of a disaccharide, plus a water molecule. This would be an example of catabolism, and we're looking here at a protein. So here you can see this is a protein, but we're going to remove the water molecule right here, and we're going to, or sorry, not remove, we're going to add a water molecule. That's what plus H2O means. And we're going to add OH, it'll then add an OH to this side of this molecule that was right here in the middle, so pay attention to this circled part. There's going to now be an OH coming off of this carbon and an H coming off of this nitrogen. So you can see them in red here. And now they are monomers, and they are separated into individual amino acids, and that peptide bond that was here originally in the circle has been broken. So an example of that in real life would be lactose, which is a type of protein. The enzyme lactase is going to add water, and that's going to split them apart right here. So now we're left with 2 separate amino acids. Oh, sorry. This is a carbohydrate example. This was a protein example up here. So lactose is a carbohydrate, and then it's going to be broken into 2. It's breaking the glycosidic bond that was here. Sometimes you might see, this is a disclaimer, you might see the word dehydration reaction whenever you're talking about a condensation reaction. Some websites use them interchangeably, but that's not correct. Technically, a dehydration reaction is when the water molecule has come from one of the reactants. So whereas a condensation reaction is part of each water molecule has come from each reactant. So an OH from one and an H from another, where a dehydration reaction would be taking it from one molecule completely, so not from 2 separate molecules. So the last topic is an application that they want you to know about for IB. So urea, which is usually something produced with your kidney, is an example of a compound that is produced by living organisms but can also be artificially synthesized. So before, we used to think of organic compounds could only come from living things, but we've found that we can actually synthesize them, so it can be man-made. So this kind of goes with the nature of science when we research. Falsification of theories is important to realize in science they can be falsified. Nothing is always 100% for sure. So what this example is it's the artificial synthesis of urea helped to falsify the ideas of vitalism. So Frederick Waller, accidentally synthesized urea in 1828 whilst attempting to prepare ammonium cyanate. So ammonium cyanate is actually an inorganic compound, and he mixed it with another inorganic compound, and it created urea, which is what we call an organic compound. So that meant it came from 2 inorganic things. So in a letter to a colleague, he said, "I can no longer, so to speak, hold my chemical water, and must tell you that I can make urea without needing a kidney, whether of a man or a dog. This is supposed to undermine vitalism, as an organic chemicals were previously thought to be synthesized only by organisms." I thought it was kind of cute how he said that, hold my chemical water. So vitalism nowadays has no credit, though, as a theory, because the idea of vitalism is, in essence, proposing that there's an unknowable factor that's essential in explaining life, which often is looked at as very religious, that there's an unknowable factor that no one knows about, and usually people were talking about God. So in the sense of the scientific theory, we don't hold vitalism as the main research or way of looking at different studies. Okay, so take a moment to see if you can answer these questions for yourself and put them in your lecture notes. So I want you to be able to answer what property of carbon atoms helps them to form such a large range of stable compounds. We're going to talk about these in class, okay? So I want you to try to guess or look back at the lecture to help you. Name 3 chemicals, or which I mean biomolecules, in living things which contain many carbon atoms. There was lots of examples throughout the lecture. I want you to state the meaning of the term cellular metabolism because so many students get it confused. I want you to really sink into your mind what metabolism is. It's not just breaking things down. So write what the meaning is. And then 4, describe the difference between animalism and catabolism with reference to macromolecules in living things. So take a moment to do that, and we will continue with 2.3 next.